Welcome back, everybody. Hopefully you found the General Session 2 tab. Hopefully you are enjoying the conference and it's working so far for everyone. Continue to put your comments and questions on the chat board. We'll do our best to keep up with them. Obviously there were lots, so we'll work hard at it. Just to set up, so this next session, 3 presentations on preclinical research. Research that happens ahead of and an attempt to validate studies that could be translated to human application. What's kind of exciting for us is already I think there may be some opportunity with these 3 presenters to collaborate together to further some of those studies and development. Look forward to these presentations. So without further ado, here's Preclinical Research Session 2. This first presentation in the preclinical research session will be from Dr. Nick Jeffery. Dr. Jeffery is a veterinary researcher at Texas A&M University, and he has been conducting a number of trials using paralyzed dogs, dogs that are clinically injured by the tens of thousands every year. Dr. Jeffery is going to additionally talk a couple of other things. One, some questions around why preclinical studies have such difficulty translating to human relevance, and also discussion of trial designs. Dr. Jeffery argues for a more pragmatic design in clinical trials as opposed to more laboratory-based explanatory design. He will raise a number of questions and make a couple of suggestions about how to improve that pathway from preclinical research to human relevance. Welcome, Dr. Jeffery.
Thank you for inviting me to present at this meeting. It's a great opportunity to meet with a whole variety of people with a whole variety of different viewpoints on spinal cord injury and research. I'm a veterinary clinician scientist, and so a lot of the work that I do focuses on problems in translation from lab to clinic, and so I'm going to be focusing on these problems, particularly how dogs might play a role in making these transitions. In February 2019, which seems a long time ago now, there was a meeting at the NIH to give impetus to a new decade in spinal cord injury research. And an interesting point was made there about the extent of the expenditure that had been made by the NIH on spinal cord injury and the lack of therapies that are being produced from that research. The research that has been done is often been in rodent models, and the result of that is that there are many successful treatments for spinal cord injury in rats. For instance, this recent PubMed search that I did shows more than 8,000 hits for rat spinal cord injury and treatment. And at the current rate of publication, there's about 600 articles per year on this subject. So the question here is, why don't these translate into clinical therapies for people? Part of the answer is that translation is just not very easy. For instance, it's been estimated that only a third of high-profile lab studies in biomedicine actually work when they're tested in clinical trials. And only about a tenth of the total of high-impact papers produce interventions that are approved for patient use. In stroke, which is quite a good comparative subject for spinal cord injury, there's been about 1,000 therapies that have been tested in animals, but only thrombolytic interventions have shown effects in clinical cases. So more specifically, why don't the spinal cord injury interventions translate? There are many, many reasons for this, but perhaps one of the most important aspects is the viewpoint that there's insufficient information regarding these interventions, particularly about the possible risk versus possible benefit that might accrue, and other aspects to do with safety. So quite a lot of people have looked at the reasons for problems with translation from lab to clinic, and a large survey was done not a few years ago by Brian Kwan, where he asked a number of researchers about the reasons for making progress in the next steps towards clinical application. And the data that's presented here is asking a specific question about what the next step should be after demonstrating that a particular cell transplantation improves recovery in rats. And so, as you can see here, there's a slim majority in favour of getting more information by applying the same therapy in a large animal model. So in summary, the gap is currently too wide to bridge between what we know where we are with rats to where we want to get to with people. So the question that I'd be posing here is, maybe dogs can help. And the reason that they might be helpful is that they can provide an intervening step to make the leaps between one species and another a little bit smaller, in the same way that this bridge in the further fourth bridges over this larger gap. In dogs, we can measure limb coordination, we can measure bladder function, and we can measure spinal cord conduction pretty straightforwardly. And there are many other measures that we can also take that might be relevant for human spinal cord injury as well. In fact, we've already translated some of the successful therapies that have been used in rats and applied them in dogs, in clinical trials in dogs. For instance, this first study, we used olfactory and sheathing cell transplants that were derived from the dog itself and then transplanted back into the chronically damaged area of the spinal cord. And the result that we got from this was that there was an improvement in the coordination between the front and the back legs. On this graph here, a lower score indicates better coordination, and you can see there's a gradual improvement in the coordination in the OEC recipients and a gradual probably worsening in the control dogs. And so of the two groups, there was a definite impact of the olfactory and sheathing cell transplants. However, there's a bit of a problem with the olfactory and sheathing transplants in that it's a slightly cumbersome treatment. We need to harvest the cells from the back of the nose, then we need to put them into culture for about 2 weeks before we can transplant them back into the spinal cord injury. So we thought that it would be useful to look at another intervention, and the one that was quite a front runner for possible translation at the time was chondroitinase ABC, and so we injected this into the spinal cord of chronically injured dogs in the same way that we'd injected the olfactory and sheathing cells, and carried out a second randomized controlled trial in dogs. And again, this showed improvement of the coordination between the fore and the hind limbs, as shown in these three graphs here. So the control animals, there was little change overall between baseline and 6 months. You can see here that there's a improvement in the group, in the chondroitinase group at 6 months compared to baseline, and that's shown in this graph here, where there's a gradual improvement in function with time. As I mentioned before, the lower score indicates a better coordination. In addition to overall group effects, an important aspect of translation is the magnitude of effect in each individual, and also the proportion of individuals in a treatment group that get meaningful benefit. These are both important questions that are likely to be asked by future recipients of the treatment were it to become approved for use in people. Patients are likely to want to know, how much impact is this going to have in my life, and what is the chances of me getting that impact when it's given to me? Fortunately, we can also answer both of these questions in dogs. For instance, in this graph here, investigating bladder compliance against the pre and post time points, we can see that most of the animals in this study, there was a little bit of improvement, but maybe not enough to make much difference to anything that would be noticed by their owners. But one dog did show a marked improvement, and that might be enough to really have an impact on the way of life for the dog and for the owner. So when we completed these studies some years ago, we thought that the results were quite strong, that they showed quite a strong effect of both olfactory and sheathing cells and the chondroitinase in improving the outcome for the affected dogs. And we were thinking that that might provide some impetus to go forward to do clinical trials on one or both of these interventions in people. But that just hasn't happened. And so we began to think about why that might be. So there might be several reasons for that, but perhaps one of them is that we focused on an outcome that was not of much interest to the patients that might want treatment, and also to the clinicians that are deciding what treatments might be valuable. For instance, although we showed that there was an improvement in the coordination between the front and the back legs, we only showed a transient response in terms of bladder function. So the compliance, the ability to retain urine in the bladder, seems to have improved a little bit following the chondroitinase therapy, but this isn't maintained over the full 6-month period of the study. And so perhaps there's some disappointment that although we could show improved coordination between the front and the back legs, we weren't able to show long-term effects on bladder function, which was perhaps regarded as a more important outcome. Another aspect is perhaps that the outcome that we were looking at was too technical. And by that, what I mean is that we were looking at the coordination score between the front and the back legs. And although there's quite a strong group effect here, so that the chondroitinase treated animals have significantly improved function, perhaps it's difficult to understand what that means in everyday real life. And this is a problem just generally in spinal cord injury research that can be leveled at rat experiments as well, that if we look at the behavioral outcomes that show an impact of a particular intervention, it's difficult to know exactly what that means if that was to be translated into a human patient. How much impact would it really have in their everyday life? And so there's still a gap to be bridged here. We've got the intermediate model of the dogs, but we still haven't quite filled this gap to show enough information to be able to bridge it, to show the information that we need to be able to justify human clinical trials. So more recently, I was wondering whether perhaps a pragmatic trial might break this deadlock. This table here is meant to illustrate a number of the features that can be used to differentiate between a pragmatic and an explanatory trial. So pragmatic trials on the whole have outcomes that are much more relevant to participants in a trial. For instance, the longevity, quality of life, and in this context, perhaps the ability to walk, the ability to control urination. And so what we could do would be to rerun our clinical trials in dogs, because it's relatively cheap for us to do this, especially with very pragmatic outcomes. And then what we need to do is to devise real life outcomes that are much more convincing in terms of showing an impact on the everyday life of these dogs. So for instance, we could have a dog walking in a park and just monitor the amount of times that it fell during a particular distance of walking. So if we were to do a pragmatic trial in dogs, this is what it might look like perhaps. So we would have the population that we would select for the trial would be severely affected dogs. We could use a number of interventions. It could be acute or chronic injury. Here I've just mentioned durotomy or duroplasty because there's been quite a lot of interest in this in both human and veterinary medicine. The outcome here, I'm suggesting that perhaps we could say that there were no falls over 35 meters when the dog was walking, and we could have that as a yes/no answer, or we could perhaps look at it as the number of falls over that distance. Here I've worked out a sample size calculation for this study where we could expect 50% success at 6 months in controls and maybe 70% in the intervention group. And we'd need 91 dogs per group to be able to do that trial. And this is extremely doable in veterinary medicine. We see a very large number of dogs with spinal cord injuries. So the big question here is whether if we were to show that level of success, that the intervention that we chose was able to create this improvement in falls over the distance that we're interested in looking at, would it lead to a human clinical trial? Would it provide impetus to carry out a human clinical trial? And I guess that's the question that I'm posing both to myself and to anybody else who might want to ask me about it. So lastly, I'd just like to thank the people that work with me on these studies that I've presented. First, Nicholas Granger, who worked with me on the olfactory and sheathing cell transplants when I was working in England. And secondly, Hilary Hu, who worked on the chondroitinase project here in the USA. So thank you for your attention, and I'll be happy to answer any questions. Thank you.
Chondroitinase ABC, or CHASE, is a naturally occurring enzyme that's long been studied as a potential therapeutic for spinal cord injury. The trouble is that it's not a very stable enzyme in its wild form, natural form. It doesn't have a very long shelf life. It's not very long. Its activity in the body, not very long. And there have been challenges in terms of how to deliver it to the body. Many of you know that, have followed chondroitinase ABC over the years. What you may not know is Dr. Molly Shoichet and her team from the University of Toronto have recently published what appears to be a more stable version of CHASE, both on the shelf and in the body, having a lengthier active life in the body and a delivery mechanism. Dr. Shoichet's going to present on the science leading up to that discovery, and then the plans they have to test its efficacy now that they have tested its stability. Welcome, Dr. Shoichet.
Hello, my name is Molly Shoichet, and I will be presenting our research from the University of Toronto on degrading the glial scar to promote regeneration after traumatic spinal cord injury. The researchers who have done this work are highlighted below, and those in blue are our close collaborators. We are inspired by biology in designing polymers in treating either cancer or in regenerative medicine. And it will be our work in regenerative medicine and specifically on spinal cord injury that I'll focus on today. And in the context of regenerative medicine, we're focused on both cell delivery and drug delivery to stimulate the endogenous cells. And here, some of the key questions we're trying to overcome are cell survival, cell integration, and plasticity. I thought I would introduce you to our research in spinal cord injury with this short video.
Spinal cord injury is devastating. It can result in permanent damage to the spinal cord's ability to send and receive messages. At the cellular level, there is massive disruption to axonal networks. Many neuronal and glial cells die, and an aggregate scar is formed. Apart from standard rehabilitation, there are no treatment options. The Shoichet lab is developing an interdisciplinary approach comprised of stem cell transplantation, biomaterials, and scar-degrading enzymes to promote spinal cord injury repair. To replace the cells lost due to injury, we have identified a population of stem cell-derived neurons that form long processes in the spinal cord after transplantation. However, ensuring cell survival and integration into the existing neural circuitry remain significant challenges. To promote the survival and integration of transplanted cells, we have engineered a hydrogel using a physical blend of hyaluron and methylcellulose that is easily injectable, enables biomolecule delivery, and is bioresorbable. Once injected, the hydrogel cross-links to form a minimally swelling gel. Extracellular matrix-derived peptides can be conjugated to the hydrogel to promote cell adhesion, thereby further improving cell retention and survival. However, another challenge remains. These newly transplanted cells need to integrate with existing neural circuitry. We at the Shoichet lab have been looking to scar-degrading enzymes to modify the cellular microenvironment and enhance the integration of the newly transplanted cells. Chondroitin sulfate proteoglycans are potent inhibitory components of the aggregate scar. They prevent axonal regeneration, synapse formation, and plasticity. Importantly, these can be degraded. To enhance integration, we at the Shoichet lab have engineered a more stable and active form of chondroitinase ABC and developed a strategy for targeted delivery. While these interventions are still being tested in preclinical models, there is hope that one day these strategies of stem cells, biomaterials, and scar-degrading enzymes will provide the foundation for spinal cord injury treatment.
So that video was made by Lesia Sazaika. So let me tell you about some of the data behind that video. One of the things that we realized, and really anybody who works with chondroitinase ABC will realize, is that it's a very fragile enzyme. And the standard methods that we had developed and others in the field have used to control the release of drugs or proteins just would not work with chondroitinase ABC. So we invented a new strategy to control the release of chondroitinase ABC, and this was invented by Katarina Volek and then advanced by Małgosia Pokulska. And in this strategy, basically what we're taking advantage of is a series of on-off interactions as a way to control the diffusion of a protein through our hydrogel. So what you see in this image is our hydrogel, which is loosely cross-linked, and those are the blue strands. We've modified that with a green dart, and the green dart is a circumology 3 or SH3 binding peptide. And that SH3 binding peptide has a certain affinity for circumology 3, which is now expressed as a fusion protein with our chondroitinase ABC. And that is shown as the yellow dart embedded in the red semicircle. So by controlling the affinity between the green and yellow, that is between the peptide and the protein, we can control how quickly or slowly our protein of interest, which is chondroitinase ABC, diffuses through our hydrogel. And the data that demonstrates that idea works is shown here. And if I can draw your attention to B, which is in the upper right, what you can see is the diffusion of chondroitinase ABC through our hydrogel, which is just methylcellulose, and that is very fast, and that is shown here by the circles. And then you see that if we have a weak binding peptide, so a weak interaction, we slow the release down, and that's with the diamonds. And if we have a strong binding peptide, so a stronger interaction, that slows down the diffusion further, and that's shown with the squares. And as you can imagine, by analogy of driving down the road and having to stop at stop signs, the more stop signs you have, the slower your progress will be down the road. And the same thing is true if we have a greater number of peptides immobilized on our hydrogel, we will also be able to slow the diffusion of the protein, and that's shown with the triangles. Now, importantly, if I draw your attention to the bottom right, the chondroitinase ABC that has been released from our hydrogel remains active over 7 days. So having now developed a way to control the release of chondroitinase ABC, we wanted to look at a way of delivering it in situ to the injured spinal cord. And we invented this strategy several years ago, where we incorporate our protein of interest in a fast gelling material, a hydrogel. We can inject that through a very small syringe needle under the dura to the subarachnoid space. And that will then, the hydrogel will localize our protein wherever we inject it. And this is, in fact, a strategy that we've used repeatedly now for several years. With chondroitinase ABC, we were able to detect it in the spinal cord over 28 days, which you see in the top left corner. And looking at the depth of penetration into the injured cord, we can see evidence of chondroitinase ABC at 2, 7, and even out to 28 days. Now, in terms of efficacy, we were looking at degradation of the chondroitin sulfate proteoglycans in the injured spinal cord. And we see evidence of degradation at 2 weeks after delivering the chondroitinase ABC, which is shown in the green, the light green bar, significantly less CSPGs versus vehicle alone. And that was most prominent at 2 weeks, but not at 8 weeks. Well, one of the things we wondered is, could we make our chondroitinase ABC more stable by point mutations? So we looked at making 4 individual point mutations in the chondroitinase ABC. This work was done by Marian Hiderachi in collaboration with Matthew O'Meara. And Matt did the computational modeling to predict which single mutations would provide more stability. And then Marian tested this experimentally and also looked at modifying this protein with polyethylene glycol. So I'll draw your attention to point mutation number 1, because this one, where we convert an N to G, an asparagine to a glycine, that was the most effective of the mutations. You can see that data right here, where that's mutation number 1, which is red, and it's significantly more stable than our starting, which I'll call our wild type chondroitinase ABC SH3 fusion protein. We see that significant difference at later time points at 12 and 24 hours. The other question we had is what would happen when we did a modification with polyethylene glycol. So we looked at mutation number 1, modified it with polyethylene glycol, and again we see greater stability of this mutation. It starts off actually initially being more active and maintains greater activity at 24 hours and even out to 48 hours. Now, as excited as we are about this research, I do acknowledge that we have lost 90% of the activity out to 48 hours. Notwithstanding, looking at injecting this onto the brain tissue in a stroke injury model, we looked at the ability of this fusion protein, this mutant fusion protein, to degrade the glial scar. And we saw greatest evidence of reduced CSPGs in the perilesional area, both 100 microns outside of the lesion area, and these areas are shown in A above. and also in the 100 to 200 micron area after 14 days. So our next question is could we do better? What happens if we're not just looking at single mutations, but what would happen if we were looking at multiple mutations? So Matt O'Meara went back to his computational models using PROS and Rosetta, and then predicted 3 different mutants. Now this has either 37, 55, or 92 different mutations shown in A, B, and C respectively, with the red balls in A, the red and orange balls in B, and the red, orange, and yellow balls in C. So Matt did the computational modeling, and then Marion, again, did the experimental. And what we can see is that all of these mutants now can be expressed as fusion proteins, and they are all more stable than our starting wild type material shown in black in B. So we know that they're more stable because we get a higher protein yield. And also in D, when we look at the temperature at which we're going to see denaturation, that has also increased for all the mutants relative to our wild type, which again is shown in black. So not only are the mutants more stable, but they're also more bioactive. And we looked at bioactivity by degradation of chondroitin sulfate A and dermatan sulfate, and you can see that the activity of the red one, which is the one with 37 mutants, is not only more stable, but also more active, and also more active after 7 days. And because it's more active, we also see that it can degrade more of the chondroitin sulfate, which is shown in C. And while the half-life of the green one, the 55 mutant, is greater than that of the 37 mutant shown in B, because the mutant protein with 55 mutations is not as active, we decided to move forward with chondroitinase ABC with the 37 mutants. And importantly, again, we were able to demonstrate that we could control the release of this chondroitinase ABC with the 37 mutants, SH3, by taking advantage, again, of that affinity-controlled release strategy that I introduced to you previously. We can control the release shown in B, where we can slow the rate of release. We can maintain the activity. And if we look at the area under the curve in C, which is shown in D, we have significant more activity of the mutant chondroitinase ABC SH3 versus the wild type. So in conclusion, we all know that chondroitinase ABC is a potent enzyme, yet fragile. And if we can deliver chondroitinase ABC effectively, we can degrade the chondroitin sulfate proteoglycans of the glial scar, and we've demonstrated this both in the injured spinal cord and in the injured brain. And now our newly designed chondroitinase ABC is more stable, more bioactive, and can be released locally by an affinity release system. So now, going forward, our goal will be to test this either alone or in combination strategies after spinal cord injury. Well, I'm grateful to you for your attention. I'm grateful to all of our collaborators listed here, and then all the people in our lab. This picture was taken last year at a time when it was still safe to get together, and I very much look forward to that time again. Thank you.
Rounding out this preclinical research session will be a presentation from Dr. Candace Floyd. Dr. Floyd is from the University of Utah, and she will continue the conversation of the challenges facing researchers who wish to translate potential therapies to clinical relevance. Dr. Floyd will discuss the use of the pig model for spinal cord injury, and also the importance of using sex as a biological variable for investigation and further study. Maybe you see where this is going, and maybe there's a potential for some collaboration amongst these three researchers. You'll have to ask them. Welcome, Dr. Floyd.
Good morning. My name is Candace Floyd. I am currently an associate professor and the vice chair of research in the Department of Physical Medicine and Rehabilitation at the University of Utah. And I'm going to talk to you today about some of our translational research. So here's the overview of what we're going to talk about. This problem, you know, it's a lack of translation in the field of spinal cord injury. Hopefully we're going to have some collaborative tools that will help to address that. We're going to talk about those collaborative tools, which are porcine models of spinal cord injury. One of the things that we are most proud of, and I think is a really great example of how to leverage these tools, is to use assessments that we back translate from the clinic to these porcine models. The concept is that by back translating the same assessments that are used in the clinic in the preclinical models, we may increase the validity and increase the pace and success of translation. And then we're going to talk about an invitation to collaborate. So this is the elephant in the room. You all know this already. This is a very sophisticated and complex audience. So if we look at the success rates for translation of CNS drugs, including drugs targeting spinal cord injury, brain injury, stroke, and other CNS disorders, these data illustrated in these elephants, in our elephant of the room analogy, come from the FDA and come from approval data of FDA CNS drugs from the years 2010 and 2015. And what the data show is that the timeline for basic research is 25 years. So the average therapeutic spends about 25 years in basic research. The preclinical phases, on average, last about 4 years, and then the clinical trial phases, on average, last about 7 years. So as you can see, this is a very long timeline of translational discovery. And then the problem is the elephants hit a wall. And the failure rate for translation in CNS is 92%. So our idea is that we need to be involved in an interdisciplinary, active collaboration on how to improve translation in spinal cord injury research. So we really think that translational improvements will accelerate the pace of discovery and eventually lead to cures. So our overarching premise is that improved models that coupled with better rigor and reproducibility in translational research will accelerate the discovery. So here's the idea. So I'm going to give you an example of our translational research, and I'm going to use spinal cord injury-induced neuropathic pain as the example. So I want you to understand that I'm going to talk about sensory deficits in this little vignette, but understand that these same concepts apply to motor deficits, to bowel and bladder deficits. So we're interested in all of these very important clinical outcomes. So when we think about spinal cord injury and neuropathic pain, data suggests that over 65% of individuals living with spinal cord injury do suffer from some type of chronic neuropathic pain. It's often described as burning or stabbing. The pain can be constant or episodic, spontaneous and evoked, and typically you know it's characterized by being below the level of the lesion. So there's very limited success in treating this pain with common pain medications. And so there are pathophysiological changes in this pain that are poorly understood and limit our ability to translate. So we were interested in determining if our pigs, where we have induced a clinically relevant contusion compression spinal cord injury, so the pigs have a mechanism of injury that is clinically relevant, a mode of injury that's clinically relevant, and they have a recovery trajectory that more mimics human recovery trajectories than that of rodents. So in our pigs, so these are an example of paralyzed pigs. You can see they're kind of like in an upright or a sling to approximate anatomical position. So we are curious to see if there was mechanical hypersensitivity or allodynia, which is a very common measure in laboratory settings of neuropathic pain. So here's just a few examples. So we're looking at the withdrawal threshold. This is the weeks post-injury. You can see that animals in the laminectomy group exhibited no withdrawal to this neurofilament, so no hypersensitivity to neurofilament testing. But that the withdrawal threshold in these two pigs, depicted in the blue line and the red line, was significantly reduced after spinal cord injury, really suggestive of neuropathic pain. So this was very interesting to us that this monofilament withdrawal experiment yielded neuropathic pain-like behaviors in pigs. However, there was a problem with this. So this is the standard way that neuropathic pain is classically done in laboratories and preclinical research laboratories, and it focuses on reflexive withdrawal. there have been a lot of literature discussions on how this is probably not all that relevant to human pain. So in human pain, what we're really thinking about is the supraspinal experience of pain, or how the pain is perceived cognitively. So that's a really important component. So our goal was to go beyond withdrawal, reflexive withdrawal, and to measure the supraspinal component. The second confounding component in measuring pain in animals with spinal cord injury is, of course, spasticity. So it's difficult when you're only looking at a reflexive withdrawal component to discern spasticity responses from reflexive withdrawal responses. So the other thing is we need a reliable pain scale, and hopefully a pain scale that more closely mimics what is used in the human clinical situation than in the animals. So this is a picture of my collie and my little camper, and if you don't have a collie, you probably, let me just describe to you that collies have a million different barks. And so they have a bark for everything. They have a bark for a belly rub, they have a bark for everything else. So the thought occurred to me then that if a collie can communicate with me by his barks, because I'm a very good dog mom and I understand what he's saying, perhaps the pigs could communicate with us. That's my golden retriever who just gets angry if he's not included in the talk. So the concept is, can we use bioacoustics and the vocal repertoire of pigs in an experimental setting to try to have a supraspinal component? So can we essentially learn to speak pig, if you will, to understand when a pig interprets a stimulus as being painful or distressful? So this is what we did. So we basically kind of went around like your favorite documentary or your favorite docudrama director, and we took microphones and cameras and we went all around everything that pigs do in the laboratory, and we video and audio recorded them. And then we classified these recordings, and then we put them in a scale. And the idea was to mimic a scale that's used in humans, so like a 0 to 10 pain sensitivity scale. So we did that. And we came up with a scale that we call the Porcine evoked pain scale. So what's important to know is that we are administering a stimulus to the animals in this, and then we're rating their responses. So we rate several different responses for the pigs to tell us how they rate, quote unquote, the pigs are rating their pain based on their behavioral responses. So as I said, we include vocalizations. So we speak pig, and we include those vocalizations which we have associated with a pain or distressful state. We have an interruption, a pain interruption behavior. We use eating. Pigs like to eat, and so when they're eating, they're interrupted. As you can imagine, their behavior, if the stimulus feels painful to them, they'll interrupt their behavior. And we also use escape behavior. Now notice that we also use withdrawal. So we included the spinally mediated components as well. So importantly, this porcine evoked pain scale, back translated from the clinic with this idea of a 0 to 10 pain scale rating the behaviors, includes the reflexive components, as is classically done in preclinical research, and supraspinal components, like is a component of human research. So we essentially ask the pigs through their behavior to indicate the severity of the sensation that they're feeling. So that's the outcome measure. So what is the methodology that we use for the stimulus? So again, we wanted to try to back translate this from the clinic. So we use a concept borrowed from the clinic called quantitative sensory testing. So quantitative sensory testing is a method that's used to diagnose chronic pain and neuropathic pain from lots of different neurological diseases. So it determines sensation and pain thresholds. So things like cold and warm temperature, vibration testing, and what's important about QST is that, which I'll call it now, is that it's not invasive, right? So it's a thermode placed on the skin of the animal. So this is what it looks like in the clinic, and this is exactly what it looks like in the pig setting, in the pig testing setting. So there are different tests looking at thermal and mechanical thresholds. This is the analyzer that we use. I'm not affiliated with this company, except to say this is a commonly used analyzer. This is the pinprick. So notice that in the picture, the pinprick isn't an actual pin. It doesn't poke and penetrate the skin, but it's actually a stylet that pushes pressure on the skin. So what we do then is we have a pig that has a mid-thoracic spinal cord injury. So the pig is a functionally incomplete injury from the mid-thoracic level down. Injury is at level T10. And then we test dermatomes below the level of the lesion, as well as one dermatome above the level of the lesion. And so here's a series of the tests that we're talking about, but what you see from this and my other description is these are all back-translated tests in the QST parameters. And in order to maintain kind of the rigor and reproducibility of this, we use a lot of procedures to make the investigators naive to the experimental conditions. And so again, we'll have this timeline, and we use several different investigators. So people who capture the video are different from those who separate it into clips, or different from those who analyze it, or different from those who score and de-randomize it. So in this way, everyone has a part in the analysis, but no one has the complete picture. We think that reduces bias. So here's some examples of the data from the QST parameters. Again, using these scales, this Porcine and Howk scale that includes both supraspinal and spinal components of pain, what you see is that, particularly looking at this mechanical detection threshold, that pigs in this group, this includes 14 pigs, have a significant reduction or in their withdrawal threshold, suggestive of neuropathic pain. But it varies by region. And so here, again, region 1, which you see is in this area, is different than region 2. And that's important because that also mimics the clinical presentation of pain. So this is an example of loss of function when we do the pressure pain threshold test to a deep stimulus. The animals actually in the laminectomy group have a lower withdrawal response than those in the spinal cord injury group. So importantly, in this mechanical detection area, there's both a gain of function as well as a loss of function, suggestive of neuropathic pain. So in our pigs, cold did not seem to be a stimulus that was had a lot of response, but on the other hand, heat was. And so again, we're looking at the heat pain threshold to a 50°C stimulus, thermal stimulus placed on the animal. Pigs in the laminectomy group don't respond to the heat, but you can see that pigs in the spinal cord injury group by 3 and 4 or 5 weeks after injury develop a robust response. We evaluated both male and female pigs in all of these experimental paradigms. And interestingly, in this set of data, the female pigs, as depicted in the black graph here, 84% of the females had this robust pain response, neuropathic pain response to heat, where 67% of the males. So that might be important in future studies to be able to distinguish between pain phenotypes in male versus female individuals. This is a little bit of an evaluation of pain medication in the model. So IV lidocaine was administered. You see here are the administration characteristics. And IV lidocaine produced 100% analgesia in these animals. And then we tested 2 pain medications, which are commonly given for neuropathic pain. Of course, the caveat is they don't particularly work very well for neuropathic pain, and that in human populations. And that's what we saw in the pigs. So there was quite a bit of variability and limited effectiveness of pregabalin in reducing the pain or of baclofen in reducing the pain. So in summary of the presentation today, I wanted to give you a vignette to give you an example of how we're using a pig model as a translational intermediary. Our idea is that the models that have both outcome measures and mechanical characteristics of injury that are back translated from the clinic will be a bridge from discovery and perhaps rodent models or other models to a large animal model that has very many similarities to that of human to those in human. So the idea is that we have a resource that's a translational intermediary. That model and a pain assessment tools are in development. So again, motor we do, valve bladder we can do, pressure source we can do in the model, and all of these tools are available for collaboration. And as I said, we're currently developing some of these QST quantifications for other types of neuropathic pain, including post-traumatic headache and chemotherapeutic-induced neuropathy. So other tools. So the invitation at the end is I would be honored to collaborate with folks. It takes a good bit of time to set up these large animal and maintain these large animal models and these large animal laboratories. We have all the tools in place, and we'd welcome collaboration. So I thank you again for all of your time and attention, and I look forward to our discussion later this afternoon.
Well, here we are for the live panel discussion. I want to introduce you to Dr. Sasha Rabchevsky. He's one of our board members and a researcher at the University of Kentucky. And with our live panel participants, Dr. Floyd, Dr. Jeffery, and Dr. Shoichet. Take it away.
Thank you, Matthew, and welcome everybody to this discussion panel. And thank you again to Dr. Candace Floyd and to Dr. Nick Jeffery and to Molly Shoichet for all joining us in this discussion period. So I think that number one, fantastic. Thank you for putting those pieces together. Very instructive, very, very formal, and very understanding, in my estimation. I think that Candace, I like you put the elephant in the room paradigm, because I'd like to just digress for a moment to just say that Dr. Jeffery, Nick, you've been established a canine consortium for testing potential therapeutics in spinal cord injury and chosen the canine model. Dr. Shoichet, you've done some amazing, outstanding work to take an enzyme molecule that is now at the edges or forefront of clinical translation. And then Candace, obviously you've got a machine operation there that's ready to take this next step of taking translation. So I guess the big question, then we can break it down individually, is what is the biggest challenges of, for instance, taking this, if this chase mutation works out, what is it going to take for it to get to the clinical trial based on each one of your unique perspectives? And I guess, Nick, we'll start off with you with the dogs. You have a community of that seems to be underappreciated for some reasons, maybe because they're, like you said, the PETA people, or maybe not so in favorable of that. But maybe you could open up your first to say, so what would be the biggest, easiest way, number one, or hardest challenge for you, for instance, to collaborate with the other two speakers?
Okay, yeah. Yeah, thank you. The first thing is that I'd say that the dog model chose us rather than us choosing it. We're veterinarians, and so, you know, we're working from a position where we're trying to find the cure for dogs that are paralyzed. And, you know, of course, we realize that it's got implications for humans. And so it sort of, we're trying to solve both problems. The problem from my point of view seems to be a sort of reluctance to move forward from preclinical models into people. I can sort of half understand that because, you know, it's very expensive to do a human clinical trial. And it could be even more expensive for a company that's developed something that fails in a clinical trial. Can have big effects on a stock market price. But somebody's got to grasp this nettle at some stage. And I think that, you know, there's only so long you can keep putting things off and keep saying, well, there's something better coming down the line. There's something better coming down the line. The way that medicine actually really works is that you make incremental improvements, so that it's not that people have a home run that they hit to solve all the problems in a particular field. It's that clinicians make incremental advances and incremental advances over a period of 50 years. And then you look back and you go, it's amazing what progress we've made. But the first, such a cliche, but you have to make that first step. And there seems to be an unwillingness to make that first step. So I see dogs as being a way of making that first step much easier, so that we make it much more clear that we've got a candidate that is definitely acceptable to go forward into people. And I think that, you know, perhaps we've made some mistakes with the way that we've analysed dog trials that don't make them as accessible as we should make them, so that it's easier for clinicians and for trial participants to understand what they might be putting themselves in for in terms of risk-benefit ratio. And I think it's partly our fault, and that's why I'm talking about redesigning the trials to try and make that more clear.
And so again, I guess I take that back to you, Molly. Now, how you're at the edge. People are probably nipping at you, wanting to do every model right to use it potentially. Is it your estimation that, let's not even take any particular model, but let's say the rat. What would you have to do for instance, in the rat to be able to get enough information for, say, Nick or Candace to be able to be willing to take a chance with their subjects?
Yeah, so thank you so much for that question. And again, it's really a pleasure for me to be participating on this panel. And also actually just to bring to the attention to people like Candace and Nick that we've made this invention. So this is really hot off the press. We just published this paper in July, I think, of this year. So this is very new. And And we think it's actually very powerful. So the challenge that we faced with this enzyme previously was that we all knew it was potent, but we all just didn't know how to, in a sense, harness that potency for it to be useful, because it degraded. As potent as it was also very fragile to work with. So we and many other people have been looking at ways to make it more stable for a long time. And now we think we've actually been able to, like, not just incrementally, but really substantively. So now we're quite excited about that. And I think, I would love to collaborate with anybody actually who wants to collaborate with us. We know how to express the protein. We know how to control its release. We know that it will be bioactive. We've already demonstrated that in some animal models. We don't know actually how potent it will be. And so the question that I would imagine Candace and Nick would ask, and other people will ask, well, okay, so it looks really good, but is it really good? And so then that's, maybe that's start with rats just because they're more accessible, and demonstrate that it's actually has some utility there, and then, move it forward. But I'm happy to try things in parallel if others want to.
Okay, and so I guess, Candace, then in your court now, how do people approach you? I mean, like I think of the pig model, and I always think of my first introduction with Krashikov and Brian Kwan, and oh my God, no one can work with those people. But now you're sitting there wanting to. So how can people, like the other speakers or myself included, reach out to you and say, hey, I've got a particular compound?
Let's do it. So my vision that I tried to share in this, and why I'm so excited and enthusiastic to be a part of this program, was for us in my lab to set up a pipeline where people could go. And so what we have is a model. We are expert in taking high quality care of the animals. We are expert in back translating evaluations, outcome measures that are used clinically, both sensory and functional measures. Our pigs, as we talked about a little bit in the talk, and I'll emphasize here, pigs have pharmacokinetic and pharmacodynamic parameters that mirror those in humans. The skin of pigs is nearly identical to that of humans, such as it's interchangeable. So secondary complications such as pressure sores and other things that many of the folks on the call have experienced, we manage in the pigs. So we have built the machinery, as you've mentioned, and we are ready to collaborate. So all it takes is Molly saying, Candace, I really think I've got a candidate therapy. And me saying, okay, do you know some of the key factors, some of the dosing, the time windows, some of the pharmacokinetic targets that you want? If she says yes, I say, that's great. We write a grant and we're ready to go. All it takes is funding. We have been doing this with our external funding sources, such as the Department of Defense and the NIH, so that we have essentially a per pig price, so to speak. We've got it worked out that well, that we also can do these experiments under an operational standard, which is called good laboratory practice. So for those that are listening on the call, GLP is an operational standard that's required by the Food and Drug Administration for investigational new drug applications. So I also have set up the machinery, and I have become a GLP certified professional myself. So if there's a candidate therapy that's ready and there's a collaborator who wants to do it, we've got the machinery in place. We have the people and the know-how to help advance the therapy.
So to me, that's just remarkably exciting, and which is why I got Matthew and I asked you all to get together. But I want to go back to Nick now. So Nick, what about your dogs? Who's funding this stuff? If you want to do a parallel study, for instance, in the pigs, if Molly gave you some of her poison and gave some, how would this work out for you? How would you fund this?
Well, same sources actually. We'd probably be looking for NIH, DoD funding. The veterinary funding is too small, so we have to look for medical scientific funding, and it's certainly doable. We've done it before. So for a very strong candidate therapy like Molly's been talking about, I would be very happy to put that into dogs straight away. I mean, we've already used chondroitinase. We were stabilizing it in a different way, but the way that Molly's doing it is clearly, a long way down the line as being more advanced product. And so
can I ask therefore, since she's on the line, that's I guess what I was asking. You've published this, you've gotten some pretty remarkable results for this compound. Would you be willing to tell Dr. Shoichet that you'd be excited to do this? Because it seems to me that it worked, but gosh, if you could get it to work better, right?
Yeah, the place that I think the pet dogs have is that what we're doing is modeling the heterogeneity, right, that comes into people. Exactly. So, if you take a set number of experimental animals and you do your study, you can find your statistical difference, and you can find, you can do the post-mortem studies to find out what's happening. So you get the mechanism. But it's very difficult to model what would actually happen in a clinical population. And our clinical population is assessed in the same way that people are. I've been looking at the common data elements recently on the NIH, and we could tick off like about 75-80% of these, to do with MRI, different scanning frequencies, our outcome measures, the decompressive surgery that we're doing. We've got all these things that we do them routinely, actually, a lot of these things. And so we have a very large population of permanently paralyzed dogs. There's a huge population of permanently paralyzed dogs in the United States, because people don't euthanize their dogs when they become paralyzed nowadays, so they keep them alive. And a lot of those owners are willing to put them into a trial, because we've shown that there's little risk, when we've done this before, there's little risk with allodynia and stuff. And so we have I think the main thing that concerns me, that what we have to show is, does a project have a big enough effect to change an individual's life? Right. And I think we can answer that in dogs. We've been looking at some methods of analyzing data. I've been doing this in rats, but we can apply this elsewhere, about how you define what an effective intervention looks like. Like how do you find something that becomes something extraordinary, that really does change a recipient's life. And that's what we're aiming for. We're not aiming for incremental changes. We're aiming for something so that the patient gets the therapy and they go, wow, my life is now better than it's been before. And 50% of people that get this are going to get that sort of benefit. That's the sort of information that we need, I think, to make it realistic to go ahead to a clinical trial. And I think we can derive that in clinical dogs.
So Molly, what would you think your biggest intrepidation is? You've got this magic toolbox, right, that everybody wants. What is there anything that you'd be concerned about, and obviously you've got some discretion here to us what you want to reveal, but what might be the problems of putting chondroitinase into spinal cord individuals?
Well, I think the point that Nick just brought up is a really important one, because for so long with chondroitinase ABC, we saw that it was effective at degrading the chondroitin sulfate proteoglycans, right? But, and it did have some, we in our own lab have seen some tissue benefit, but not, and even some functional benefit, but very minimal. And it seems to me that no one's really demonstrated a benefit of using chondroitinase ABC on its own. And it's always been in combination to really see that improved function. So I guess our first question, and Candace, you brought up some good points in terms of, do you know everything about this molecule in terms of, pharmacokinetics and dosing? And the answer is no. Because really our first question is, now that we have this much more stable and active form of it, will it be useful on its own? Because even, to give somebody back something as significant as bladder function is really significant and would be really important. And if we could do something with one molecule, then it's much simpler to bring that as a product to people, to market as one, even if it doesn't, do everything that we want to achieve. That would be hugely significant, but we don't actually know that yet. And so that really, I would say, is our fundamental questions. Now that we have something more powerful, is it actually more useful on its own, or does it still have to be used as a combination product?
So let me jump in with a couple of questions from the audience. You've already you've been talking around the questions that have come up. But first up, sort of in line with what you just said, Molly, and to the others of you, if you'd like to chime in, two-part question. One of them is around, and to the others of you, if you'd like to chime in. One of them is around, Sasha, you might want to turn that volume off. Okay.
Yeah, sorry.
One of them is around what potential candidates for a combination therapeutic do you all think about? But then the other question came from Michael as well, which is a two-part question. There's some evidence suggesting that the chondroitin sulfate proteoglycans are not as much of a barrier as may be suspected. And then secondly, the role of the chondroitin sulfate proteoglycans in the perineuronal net, particularly in areas where you want to restore the motor neuron pools. So two-part question there about possible combination, and then this question about, well, are the CSPGs really a significant barrier? And any of you can comment.
If I don't want, I'll start because my answer is short, and I think that some of the other answers are longer. So one of the things that we've been implementing in the pig model, and I think that Dr. Jeffery probably can tell you the same thing for the dog, is a rehabilitation approach as well. So maybe not as classic a drug combination therapy, but certainly having the ability to try drug or cell therapies with rehabilitation. And it's the idea to mimic as best as possible some types of rehabilitation therapies that would be given clinically. So we are in the process of setting up, just like others have heard about, you've heard about for many years, body weight supported treadmill training in the pig model, other swimming types of rehabilitation therapies. So this concept of developing a drug therapy without rehabilitation in an animal model might also be limiting. So I think that adding functional rehabilitation training is a place that is worth mentioning.
Yeah, so I'll just add to that. I think, Candace, that's a really important point. And of course, we do have rehab for patients. And so that's really fantastic to include that as a combination strategy. We don't really do that in the context of the rats, but I think but we do test them functionally. So anyways, it's different. We've looked at both cell-based combinations and therapeutic-based combinations with chondroitinase ABC. And I would say that neither of them have been that spectacular so far. And so, I think part of it is thinking about which therapeutic proteins we would add in combination. So we've been looking at factors that will either promote axonal outgrowth or new connections to be made, so that if we're degrading the glial scar, then those neurons, those extensions can then make those connections. And then with cell-based therapy, it's, just replacing those cells, those neurons that are lost, and then trying to give them an opportunity to grow. But, so we've got some ongoing research right now with cell-based therapies that we're quite excited about, but we're still actually just analyzing the data. So I think both of those strategies are potentially quite exciting, but as I said, it does make it more complicated. I would just also point out that there's been a lot of research on not just the use of chondroitinase ABC for degrading the glial scar, but also promoting some plasticity and also influencing the immune response and the microglia. And so it's not, in a sense, even though we talk mostly about degrading the glial scar, I think there's other impacts that this molecule has, and so that it can be thought of in multiple ways.
Nick, any comments to those questions or further discussion?
I can add a couple of quick points if you like. The first one is that from the point of view of translation, I think that once you start to put other combinations in, I think it makes it much more difficult, because it's going to be difficult to design a combinational trial that's not like massively expensive, and you probably want to get one done and then the other one done, and then you add them together. So I can see there's some problems apart from rehab, of course, because rehab is a standard way of treating certainly animals. The second thing is about the perineuronal nets, is that we did deliberately target them as well when we did our chondroitinase. We did try and inject them into the sort of target area in the central pattern generator as well. So we did make 2 injections at the same time, because we were deliberately trying to break up the perineuronal nets for a short period in the central pattern generator.
Can you quickly just, as a layperson or to the lay people in the audience, explain the perineuronal net and sort of the context around that question?
The idea is that the connections that are made in the central nervous system are sort of sculpted during development, and then as you move towards adulthood, they get more fixed in the relationships that they have. And part of the way of fixing that is to have these perineuronal nets that are made of a number of substances, but chondroitin sulfate proteoglycans are one of the main components of that sort of glue down the contacts that are made between the pathways coming down from the brain and the circuitry that's within the spinal cord. But after injury, you want to start that off. You want to allow resculpting of those connections, because you want to allow new connections to be made. And so destroying the perineuronal nets as a way of opening up that opportunity for new connections to be made is, I would think, is quite an important part of this type of therapy. And that's the rationale for trying to break it up after injury.
If I could just make a comment, I think that's something that I, as a patient who went through rehab years and years, decades ago, I think that something is, to me, baffling, is that, Nick, and you mentioned it, there's a rehabilitative protocol, even for humans, although it's not standardized. If we're going to have Chase do anything, are we just going to put him into pigs and dogs and cats and rats and watch him do better? Or are we going to force them to use things? This is what I'm trying, even with human beings. I think this is something that I'm kind of, I worry that people want the perfect magic pill, and if it doesn't work, they just give up. As everybody, maybe some of you don't know in the audience, I have electronic implants in me. I have pretty much everything to try to make me walk. I'm doing biological research, but the question is that if I weren't trying to do things, nothing would happen. And so I guess use it or lose it principle, I think, applies here with Chase, if I'm not mistaken, right? If you're just going to put Chase in, do you expect the animals to get better? That's the question. If without any other intervention, whether it's obviously pharmacological, but I'm talking about a physical intervention, physical therapy to force them to do it.
Okay. So I can give you a bit of an answer on that in terms of how I think of this pragmatic trial. So the way that I would design this is that you have the 2 groups. Some of them get chondroitinase, some of them get a controlled injection. We tell the people you should do rehab, and this is how you should do it, and this are the centers that you go to do it. But we have no control over whether they actually do it. So that makes it like it would be in real life, because not everybody does everything that you want them to. And so that's an example of how a pragmatic trial would work, that you tell people that you should do this, but perhaps they won't. And so if the chondroitinase comes through that, because it's likely to make it look less effective, but if it comes through that, then it would tell us that if you put it into people, it's very likely to work, because it hasn't even been put in optimally in dogs. And if it still shows a benefit, it's likely to be truly effective and worthwhile.
Well, and I remember having this discussion with you in London 3 years back, and it still amazes me how, you know, again, I feel like the Horton Hears a Who, right? You got this model. I think it's a matter of right now, and this is why we're having this discussion, is like, let's take down the silos. Let's start to work with it. And I think you're right. Let's not be so BBB oriented and more qualitatively productive life, quality of life, which maybe even Candace, you could address in your pig models, because you can do cardiovascular outcomes.
Right.
You can look at cardiovascular outcome. And you, as a matter of fact, I think Brian invent a pig BBB or something like that.
Yeah. So yeah. I don't want to steal Molly's thunder because I'm sure she has a lot of comments on this regard, but I think you could almost see a translational pipeline, right? So in pigs, so the great news about pigs is they'll do anything for food. So pigs will do anything for a Fruit Loop or a marshmallow or whatever, right? And so you know, we could design a trial. It's a little bit more less opportunist than Dr. Jeffrey's trials, right? So we essentially could evaluate the dose of rehabilitation, right? You know, in that way, we could evaluate the dose of Molly's drug. We could look at the combination together. And then because we use bioacoustics and other outcome measures, I mentioned in my talk, I always make air quotes, we speak pig, right? So we understand the pig's interpretation of their surroundings, right? So with regard to stressful situations, with regard to, sensory input. So there are also, in addition to only looking at locomotion, which I think is a limitation in some of the outcome measures, we can look at locomotion, we can look at sensory function. Pigs have neurogenic bowel and bladder. And, you know, we look at those outcome measures. We can also look at, as you said, cardiovascular function, respiratory function, skin breakdown. So there could be a multitude of outcome measures in addition to just or merely looking at locomotion that, as Molly said, they're really clinically relevant. I mean, to me, it would maybe not be a home run, but certainly be a good, solid third base hit if we were to show that there was a significant decrease in neuropathic pain and a significant increase in some other, ADL-like outcome measures in pigs with or without rehabilitation.
Well, listen, thanks you all. Unless anybody has some last burning comments, we're kind of at the end of our live panel discussion. Very much appreciate the conversation, appreciate each of your presentations. And especially if you would like, we would love to have you join the open forum discussion at the end of the conference day, 2:15 to 3:30. You're welcome to be there. I'm sure folks that ask questions would love to ask them to you. We didn't get to all the questions and comments, unfortunately. So thank you again. Very much appreciate your time. Appreciate your research.
Thank you so much, guys. Thank you.
Thank you.
Good to see you all virtually.
Thank you. Thank you.
Yeah, thank you. Bye.
Hi there. It's Eric Howk from Portugal the Man. I want to thank you guys for letting me be a part of this year's symposium. It's an extraordinary group of speakers and a great opportunity for someone like me to listen and learn. Welcome to my little quarantine cage here in Seattle, Washington, where I've spent a lot of time with the opportunity to be able to connect with other people over things like this. So it's kind of extraordinary. And I am really happy to be here today. I just wanted to talk a minute about a song that I'd like to perform for you guys in a second, if you're a willing audience. It's a tune called Fall Bright. I think that all music and all writing in general is sort of autobiographical. It sort of has to be to some extent. Green Eggs and Ham, Ben-Hur, Find Your Truth, you know? Like, I think that there is some sort of autobiographical elements of all art. And I've never really done anything super on the nose in terms of my injury. I'm a T4 complete paraplegic, injured in about 2007, and I've continued to make music with extraordinary people and been lucky enough to have a long career traveling around in a bus, strumming a guitar. But I've never really written a on the nose song before, and this one's close. This one goes into themes about my injury and also sort of elements of my childhood in Alaska. So I could blab on about this stuff forever, but we got songs to sing and we got questions to answer. So without further ado, Fall Bright. Thanks, guys. We'll see you in a second.
Porch light on at dawn, and the power bill is absurd. For light, for bright, first fall I've seen all night. Just how do you propose? We'll get your father's truck to start. This fall light, fall bright, first fall I seen tonight. How does anybody know where the endless ends and the restless start? And this fall light, fall bright, first fall I seen all night.
Welcome back. So we are improvising a little bit because our interview guest is not on the call just yet. So we're frantically trying to find out where he might be. That would be Eric Howk from Portugal the Man.
We're going to wing it.
Giggling. So we're going to wing it. Ron, I'm wondering if we can just go ahead and play the video for, oh, actually we already did. Never mind. You just saw the video of him. My mistake. But he's not here. So Kelsey and I are going to talk.
Yeah.
Hopefully you like that video of Eric. And if he jumps on the call, we'll do so. But let me just start, Kelsey. This was your idea, really, down to it. I know we've had a bunch of conversations about this notion of spinal cord injury community. And in fact, as I've told you, actually, it's funny to me that in the last few months, there have been people who've said to me, there is no spinal cord injury community. Why do you keep using that term? There isn't such a thing. And I always respond and say, actually, I think it's aspirational. And I actually think there is a community, but we need to enforce that. We need to build it. We need to feed it. And you had this idea of, well, let's do these interviews with artists. Talk about that.
Well, I think first it was like the idea of this sort of collective, and that being this other entity of U2FP, to create more of a sense of community and have, I think for me, like there's always this balance between acceptance and hope, right? And like I have to hope for a cure, I have to strive for a cure, like that's just how I roll personally. But I also have to like live my life for today and be happy and do my thing and figure out who I am. And that's a really difficult balance sometimes. And so I think in terms of like U2FP and being involved in something and that like streamlining that through this organization, it's like, how do we have there be like something bigger that keeps us living in the now and connecting and finding joy together and creating longevity so that we don't get burnt out and always be reaching for the light at the end of the tunnel, which is, you know, the cure or some sort of curative therapies. Because as much as that's, you know, motivating and inspiring, and there's so many cool things that come out of that effort, I think an artist collective and sort of creating community that way can just like make life richer and can also make this effort stronger.
Well, tell me, so what I recall from a while back, in fact, and for those of you who don't know, I think you all probably know, but Kelsey has been working on a film project for a long time, right?
Yeah. I thought I was going to be done in like 2 years. I was sorely mistaken.
But it's coming. It's coming. So it was called the Cure Map. Now it's called Submerged. Maybe it'll be called something else.
Who knows?
Kelsey won the ITVS Grant Award, which is a PBS Independent Lens Grant Award to finish the film to guarantee its distribution. Way back when you asked me to come join you for an interview at the University of Minnesota. And it was with a professor in the public health department. He had been an active activist during the AIDS activism of like the 90s and into 2000. Yeah. And I remember him saying, this is a long slog. So one of the things that we learned in our community was actually you have to feed this sense of community. Yeah. Because otherwise you get fried and burnt out. If you're not sort of circling back some energy and life into your movement, you won't have a movement.
Right. Yeah, he was the one that sort of sparked that idea of like having a more holistic approach to whatever it is that you're striving to make change for. And I think it's really smart. You know, it just brings more joy and connectedness and makes more relationships, and I think it's a win all around. And I think this is really fun to like see what other people are doing and see how it can change you and all that fun stuff.
Yeah.
You know.
So I'm looking. So if you're looking at me like I'm looking away. It's because I wanted to see if there's any.
Are you listening?
I wanted to see if there's any questions for you. So if anybody has any questions for Kelsey, since Eric is doesn't appear like he's going to be here. Hopefully he's okay.
I mean, I could talk about the film for a second too, since you mentioned.
Yeah, please do. Please do.
You just mentioned the timing on this, and it's funny because when I started this film project, first I didn't know it was going to be about me and sort of center around my personal story. And I also didn't know that it was going to take this long. But apparently
talk about that really quickly, like that transition. So, you know, you started by, look, we're just going to traverse the United States. We're going to talk to researchers and advocates and make this movie called The Cure Map. You get further along down the road, in particular with this grant, and it's like, you have to, like, there has to be somebody, there has to be a face to tell this story. It just can't be like interview after interview after interview about this sort of abstract thing. Talk about
right. Like we had to draw people in with a personal story so that people could connect with it and see themselves in this film. And that very quickly, through like connecting with other people in film and bringing people onto our team and becoming, you know, more just like versed in filmmaking and bringing professionals in, we realized, okay, I'm gonna have to be more vulnerable here, and I'm gonna have to throw out some ideas of what I thought this film was gonna look like, which has been really hard, you know, because I want to serve this community the best way I can. And sometimes you think it's going to be one way and it ends up being this totally different way, and that's okay. But it's weird to like surrender to that and to like adjust to that, because like I also have had to just like really open up with a lot of things about my life and who I am and put myself out there. And that's kind of scary. And it's also kind of scary in terms of like all of this work that we did going on the road. You know, we drove around the entire country for 2 months and interviewed like 80 people, I think. And you just can't have like 80 talking heads out of film. It's going to be like so boring and stupid and all over the place.
Kind of like We're Gonna Walk, you mean?
Just kidding. I did not say that. But yeah, like it's interesting to become a storyteller and to like really figure out what's the best way to do that, and what's the best way to tell people, hey, this is what this person, being me, which is weird to look at yourself objectively, this is what this person wants, this is what the community wants, this is this story, and how can this film affect people to make them see the change that we want on so many levels, and what it's like to go through this, and how can they see themselves in this film in order to really feel something, you know?
Well, to also comment also that I mean, it's funny to me, the way we've talked about this in the past, you know, for anybody who's read I don't even know what field this would be in, but like this idea that consciousness, you know, or that your consciousness is sort of a trick of the mind, like that you're this fixed thing, you know, and one of your recognitions I know we've talked about is like over the time, and you look back, you realize, oh, I'm different today than I was 2 years ago, or, you know, when I started and what I was talking about and how I was framing it, I'm different than I was even just back then. And how that's influenced the sort of construction of the narrative and the story that you're telling, and what a cure means to you and what being happy means to you, all that kind of stuff, right?
Yeah, I've evolved so much during this process as a person with a disability. Like, I didn't even say that. I didn't even call myself disabled when I started this film. I wasn't even there yet with my injury, where I was comfortable saying I'm a person with a disability or I'm disabled. Like, that was still felt like I just didn't know yet what, you know, that didn't have to be a bad term. That's like, you know, like I can feel empowered by saying that. And there's just been so many changes throughout this process where I've, yes, I still want a cure. Absolutely. I'm always going to want my body back. But I'm also really evolving as a person with a disability and starting to like see a lot more of the whole picture, you know, of what I want and who I am and what this whole community, I'm not going to speak for the whole community by any means, but I'm going to do my best to like show people what we deserve and what we want on a multitude of levels.
And certainly another thing we've talked about a lot is you want people to respond, right? So you want to invite people into this work, you know, again, similar to why we do this conference. You know, we want to engage people from our community, keep calling out our community to get involved in this work. That's kind of what you want for people who see the film. When can people see it? Where are they going to see it? How are they going to see it?
Oh man, I wish I knew. So right now we're working with our editor, we're working with a composer, which is really fun and exciting. And we're working on our first rough cut actually of the film. So I've seen an assembly of the film, but I think the final cut will probably be done in March. And then from then on, with COVID, I don't know exactly how distribution is going to work and festivals and all of that. And the timing from there is kind of from fine cut, delivering that to actual broadcast, like being on one of ITVS's streaming services. I'm not sure about that timing, but I'm going to keep you guys updated and follow me and the film on Instagram. It's Submerged Film, and I'll definitely keep you all posted for sure.
Great.
Mm-hmm. So I'm pumped. It's
looking forward to. I've seen little snippets of it. Looking forward to seeing the whole thing.
Thanks, buddy.
And hearing the music and all of it. Yeah.
Yeah.
Really cool.
It's pretty exciting.
And
your son's going to be in it too, which is cool.
My son's going to be in it.
Yeah. And you're in it. I'm not going to give any more spoilers, but okay. Yeah.
Well, I'm grateful to not have yet hit the editing floors chopped off.
No, you're not on the cutting room floor. You're a star.
Whatever. Well, listen, thanks. And thanks sorry that to you all in the audience that don't know, haven't heard back from Eric. We didn't make this whole thing up, obviously, because he did, put together the video for us. Maybe we'll be able to incorporate somewhere later into the conference. Appreciate you, Kelsey. Appreciate this idea for these artist interludes. And of course, there's 4 more to come, both Friday and Saturday with a number of different artists and different media. So we'll continue that conversation.
Yeah.
Thanks.
Thanks, buddy.
Yeah. Okay. So to the rest of you, that's the end of our first conference day. Very much appreciate you being here with us. Just a couple of things to remind you of. One, there are 3 meetings that you'll be able to join. You can go to the virtual lobby, into the breakout rooms. We have invited the scientists who presented today to join them. Don't know if they'll be able to make it or not. Hopefully some of them will, so you can ask questions of them there. Remember our kind of admonishment. Make sure to kind of watch yourself, mute yourself if you're not talking. With a lot of folks on a Zoom call like that can get a little, crazy. So that's number 1. Number 2, you can only join those conference rooms in the Zoom call, so it's not on the live stream platform. We'll have to exit this and go to that Zoom call to join those meetings. And then that open meeting will start again tomorrow morning at 10 a.m. Central Standard Time, where we'll start the conference over for the rest of the sessions tomorrow. And then lastly, if you had any issues today, or if you know of anyone who had any issues with the technology, please email us at unite, UNITE, at u2fp.org. And we'll hopefully take care of that for you between now and tomorrow morning. Thank you so much for being here. Appreciate your attendance, appreciate your participation on the chat boards. Hopefully you have a great either networking or conversation post now that'll start right away. Thank you. See you tomorrow.